Hello, dear students. I welcome you all on behalf of Sri Jendra Rank Zone classes. Dear students, we are in continuation of the plant breeding part of this chapter, that is strategies for enhancement in food production. Okay, and in plant breeding, what is the technique? How plant breeding is done? Okay, and there are some related things with that. That is green revolution. That all thing we have, or in semi varieties. That all thing we have discussed in the previous lecture. Today, we are going to make a breeding of a plant for the what a disease resistant, because yield can be improved by the cemetery of varieties. But to increase the disease resistant, there are some hybridization or some techniques carried out to improve the disease resistance. So, if we talk about a plant breeding for disease resistance, a wide range of fungal, bacterial, and viral pathogens affect the yield of a cultivated crop disease. We know that there are fungal infections, bacterial infection, viral pathogens, which ultimately infect the plant and decrease its yield. Okay, and specifically it occur in the tropical climates. So, if we talk about such symptoms on the plant due to these infections, these are some of the diseases, or we can say bacterial, fungal, or viral diseases. And due to this infection, crop losses 20 to 30%, up to 20 to 30%, or sometimes even total loss can occur. So, in this situation, breeding and development of cultivars resistant to disease is these diseases which are developing due to the some of the particular pathogens. So, to resist against these diseases can be developed in a new cultivar by the particular breeding and development programs, and which can lead to the enhancement of the food production. So a race or variety of plant that has been created or selected intentionally and maintained through a cultivation, that is cultivar. Cultivar is what? A race or a variety or a new plant that has been created and selected intentionally and maintained through the cultivation. On a regular cultivation it is used. So breeding is done like this. We can say in this particular diagram, it is a genetic engineering which is shown here. Actually, it is not breeding; it is genetic engineering occur like this. And the hybridization, we have studied that hybridization is done by the two different varieties of plant with the desired characteristic and make a hybrid in the which will produce a characteristic in the new generation. And these are the cultivars. Okay. So this also help to reduce the dependence on the use of fungicides and bactericides. So if we create already a plant which is resistant to the disease, then the use of these fungicides and bactericides will be reduced or decreased. So the resistance of the host plant is a ability to prevent the pathogen from causing a disease. Okay, so it is a resistance can be developed in a plant. Then it is called as a disease resistance, and it determined by the genetic constitution of the host plant. Host plant's genetic constitution can determine. Whereas before breeding is undertaken, it is important to know about the causative organism of that particular disease and the mode of transmission. If we know the causative agent and the mode of transmission, then it is possible that we can identify the way or modification in the genotype of the plant, so that will help to gain a disease resistance. Okay. So now here we know that pathogen is an organism that causes a disease in another organism, that is called host, and there is a too much reduction, that is 20 to 30% even total. So it is known that the development of, we can say, development of disease in a plant depends on the interaction among following factors. Okay, so development of disease in a plant depends upon the following factor, and that is host genotype, then pathogen genotype, then environment. Now there is a continuous interaction of host genotype with the plant genotype, pathogen genotype, and with the environment and with the with the pathogen. All these three are in a interaction. Now, if we take a host plant, and that is the particular pathogen, and there is an environment. If we modify a pathogen and if we gain a disease resistance, then it will be for a short period of time, because the genotype of pathogen will be modified and again it will be susceptible for the infection. So environment also we can't notice. So there is a change in host genotype will be more preferred to gain a long-term disease resistance. So it is known that we cannot exercise control on pathogen genotype, because it keeps on changing. It keeps on changing with time. Now it is known that some hosts possess the ability to prevent a pathogen strain. Some host plant or host genotype possess or have the ability to prevent pathogen prevent pathogen strain from producing disease. And such hosts are called resistant to disease, or resistant to the pathogen. Whereas this ability is the resistance. So resistance is the host plant of the host plant is the ability to prevent a pathogen from causing disease, and it is determined by the genetic constitution of the host plant. So resistance is determined by genetic constitution of host plant. Okay, so this is the thing which is observed. Now it is known that some of the plant species caused by fungi, bacteria, and viruses, and we are going to discuss that particular disorders which are caused by this different pathogen. So some of the following plant diseases are, and the causative agent of them are. Suppose the fungal diseases are there. Some of the fungal diseases are there, which are caused by the in plant by a fungus, different fungus. So if we talk about such fungus, then the disease which is fungal disease is a brown rust of wheat. Now this brown rust of wheat, if we talk, another is the red rot of sugar and another is the late blight of potato. Okay. So if we talk about this brown rust of wheat, this brown rust of wheat, it is caused by Puccinia recondita. Okay. Same way, red rot of sugar cane, if you talk in sugar cane, it is caused by Colletotrichum falcatum, and the late blight of potato, it is caused by Phytophthora infestans. Okay, so such characteristic as you see in brown rust of wheat, you will see the brown rust like characteristic. Then red rot is such a way it is observed, and late blight of potato whether get or potato get damaged. Okay, so that is caused by different this species of the fungus. These diseases are caused. Apart from this, apart from this, some other fungal diseases are like loose smut of wheat. Now this loose smut of wheat, if we talk, it is caused by, these are the name of this is in the, here we are going to talk about causative agent. So if this is a fungus, then this fungus is Ustilago nuda treatise. Ustilago nuda treatise is the causative agent of this, that is known as loose smut of wheat. Okay. After this, there is a black rust of wheat is there. Now this black rust of wheat, if we talk, it is caused by Puccinia graminis treatise. Puccinia graminis treatise causes the black rust. Then it is known that yellow rust of wheat, this yellow rust of wheat, it is caused by Puccinia striformis. And this Puccinia striformis, it is also known as Puccinia glumerum. Apart from this, the another particular fungal disorder which is caused, that is, so these are all the fungal disorders, okay, which are caused by the fungus. Now after this we are going to talk about the bacterial disorders. So bacterial diseases which are caused by the bacterial strains or some of the bacteria. So if we talk about such examples of the bacterial diseases, then they are the black rot of crucifers and the citrus canker. So this is the black rot of crucifer and this is the citrus canker. So in black rot of crucifers, the crucifers, or we can say this is the cabbage get black colored, and this like this coloration is lost. In citrus cankers, the citrus fruits are affected. Now this black rot of crucifer, it is known that it is caused by Xanthomonas. It is caused by Xanthomonas campestris. Campestris. Okay, so that causes a black rot of crucifers. Apart from this, the citrus canker, that is caused by Xanthomonas citrae. That is going to cause a citrus canker. Okay. Apart from this, if we talk about the bacterial disorders, it is known that the another bacterial disorder is red stripe of sugarcane, and this red stripe of sugarcane is caused by Pseudomonas rubrillians. Okay. Apart from this, if we talk about the another disorder in rice, so that is bacterial blight of rice, and this bacterial blight of rice is caused by Xanthomonas oryzae bacteria. Okay, so these all are the bacterial disorders. Then the viral pathogen which causes some of the viral diseases, if we talk, the example of the viral diseases in tobacco, it is a tobacco mosaic virus, and in turnip, it is a turnip mosaic virus. Okay, so these are the diseases, tobacco mosaic and turnip mosaic. So this is the tobacco mosaic, and this is the turnip mosaic. Now it is known that this tobacco mosaic is caused by TMV, that is tobacco mosaic virus, and this turnip mosaic is caused by turnip mosaic virus. And this turnip mosaic virus have single stranded RNA. Okay, so these are the viral diseases. So these all diseases are caused by the different viral, bacterial, and fungal pathogens. Now, after understanding this, we are clear that the disease resistance against some of the pathogens which are causing the disease is required, and it can be obtained only by the changing in the host genotype. Okay, so now we are going to talk about the methods of breeding for a disease resistance, by which methods we can gain a disease resistant in the plant pathogen. So incorporation of disease resistant in an improved variety is the cheapest and hazard free means of controlling disease. It is the cheapest method, the hazard free method to incorporate a particular disease resistant in the improved variety. Rather, it is only means for a predictable control of many diseases. So breeding for disease resistance is carried out by a conventional breeding technique or by the mutational breeding. So breeding is carried out by conventional breeding, and in this conventional breeding, the method of breeding for a disease resistance is that of hybridization and selection. So hybridization and selection is the conventional breeding method. And another method is the mutational breeding. Okay, either by the conventional breeding technique or mutational breeding, the disease resistance can be gained. Okay, and mutational breeding can be like this. Okay, so the steps are essentially identical to those who are breeding for any other agronomic character such as high yield. The steps are similar. So the various steps involved in conventional breeding, as we have discussed in the previous lecture, first is the screening of germplasm for the resistant sources. We have to screen the germplasm. We have to identify the particular seeds which have which seeds of the plant which had a resistance to the some of the diseases. Then hybridization of the selected particular parent, that which has a disease resistance, that have to be selected as a parent. Then selection and evolution of the hybrid. After hybridization, we have to evaluate the hybrid, and after a particularly superior recombinant we get, then there is a need of testing a release of the new cultivar. So this way particularly the steps are carried out. So some crop varieties bred by hybridization selection or by conventional breeding for a disease resistance to the fungal, bacterial, and viral diseases are. So some varieties have been developed, okay, through the hybridization and selection or conventional breeding program. And these varieties are, if we talk, this table shows the particular varieties which are produced by the conventional hybridization, and they gained a disease resistance against the fungi, bacteria, and viral diseases. So if we talk about this table, this is the NCRT table. We remember this is one of the important table because so many times in Neet they ask direct question from this table. Okay, so this the tables, 2 to 3 tables are there in this particular chapter, or in a disease resistance and base resistance, that you should remember. So in wheat they have developed a new variety through the conventional breeding, that is Himgiri. So Himgiri variety of wheat show a resistance against some of the diseases like a leaf and a stripe rust and the heel blight. Now leaf and stripe rust means the rust is caused on the leaf part, and heel blight means breaking of the tip part. Okay, so against such disorders, the resistance gained by the Himgiri variety. In Brassica, also there is a one variety that is named as Pusa swarnim, which is also known as Karam rind, which gain a resistance against the white rust. Then in cauliflower, there is a two varieties, Pusa subra and Pusa snowball K1. This both variety, if we talk, this they gain a resistance against the black rot and the curl blight black rot disease. Okay. Black rot means black colored dark patches will be developed, and color blight black. As same in the culling in the leaf occur, and the blight and the black rot is developed. Same way in cowpea, there is a Pusa komal variety which is against the bacterial blight disease. And we have studied just now that bacterial blight in rice is caused by Xanthomyces oryza. And here cowpea is there. So here bacterial blight disease is resistant variety is Pusa komal. In chili, the resistant variety against this is chili mosaic virus, tobacco mosaic virus, and curl leaf is the Pusa sadabahar. Okay, so it is an important table. You have to mug up it, but direct question from the in a need you can get from this particular table. Okay, so Himgiri variety which we have studied of wheat is resistant to the leaf and striped rot and hilbert we have studied. Okay, then Pusa surname or Karan rice just we have said against the white rust. Then Pusa subra and Stobol came a variety that we have understood against the black rot and cull black rot. And Pusa komal variety of cowpea is against bacterial blight. And Pusa sadabahar is the last, which is a chili that is against the chili mosaic virus, tobacco mosaic virus, and leaf curl. Okay, so these are the varieties which are developed by the conventional breeding. Now, conventional breeding is often constrained by the availability of the limited number of disease resistant genes. Okay, conventional breeding in this we can get a limited number of disease resistant genes in the project and that are present and identified also in various crop varieties and wild these genes are limited. So, okay, so now several other examples of this are there, and in that several wide relatives of the different cultivated species of plant have been shown to have a certain resistant character but have very low yield. Okay, so if we talk about here several wild relatives of different cultivated species of plants have been shown to have certain resistant character but have very low yield. Hence, there is a need to introduce the resistant genes into high yielding cultivated varieties. Okay, and if we talk about the example, then it is known that let's write this example in the next slides. It will be easy. Now it is known that the resistance to yellow mosaic virus was incorporated in bindi that is Abelmoschus esculentus. Okay, so resistant to yellow mosaic virus were incorporated in the bindi that is Abelmoschus esculentus from a wild species that is Abelmoschus many hot. And it is known that the new resistant variety of Abelmoschus esculentus is called Parvani Kranti. Okay, so this is the particular new variety of Abelmoschus esculentus was developed. But this is all a method of conventional breeding. Now, we have just studied that conventional breeding is often constrained by the availability of limited number of disease-resistant genes that are present and identified in various crop or relatives till now. So now there is an inducing mutation in a plant through the diverse means and then screening the plant material for resistance subsequently to the desirable genes being identified. So now the next method is of a mutational breeding. So inducing mutation in some plant through diverse means and then screening the plant material for the resistance sometimes led to the desirable genes being identified. So plants having desirable characters can then be either multiplied, means whatever the offspring we get through the particular inducing mutation that have that either multiply directly or used in breeding. Now, other breeding methods that are used selection amongst the somaclonal variants and genetic engineering. Apart from this, uh, in mutation, conventional breeding and mutation, these two are important, and that we will discuss what is somaclonal variant method and genetic engineering, how they are useful in gaining disease resistance. So mutation is, if we talk, it is a process by which a genetic variation are created through change in the base sequence within a gene. Okay, what is the base sequence or nucleotide sequence within the gene get changed, and that result in a creation of a new character or a trait not found in parental type. It results in a creation of a new character or trait which is not found in the parental type. Okay, so now it is a process, okay, through which this genetic variation are created and the base sequence are changed that result in a character of new variety which is not found in the parental type. So new genes are identified. Now mutation, if we talk, it is possible to induce mutation artificially through the use of chemical or radiation like gamma radiation and selecting and using the plant that have a desirable characters as a source in breeding. So we can provide a mutation artificially by using this mutagens, okay, that are like the chemicals and the radiation. So this process is called mutational breeding. So by inducing a mutation, we can create a new variety. So now, if we take examples of this, in mung bean, it is known that resistant to the yellow mosaic virus and powdery mildew were induced by this mutation. So it is one of the important example of mung bean where induced mutation helped in incorporation of this resistance. This is the mung bean. Several wild relatives of the different cultivated species of plant have been shown to have a certain resistant character but have very low yield. Okay, so hence there is a need to introduce resistance genes into high yielding cultivated varieties. And this can be achieved by the mutational breeding. So now if we talk about the mutation are of two types, we can say. So how this mutation can be induced and a new variety can be gained. So mutation occur in nature at a very slow rate. At a very slow rate. These are called spontaneous mutation. Okay, so mutation occur in nature and they are very at a very slow rate. These are called spontaneous mutation. They are occur automatically. So, in the view of the extremely low frequency of desirable mutation, spontaneous mutation cannot form the basis of the breeding program. Okay, so in view of extremely low frequency of desirable mutations. Desirable mutations, spontaneous mutation cannot form the basis of a breeding program. Okay. Now, so there is a need of induced mutation, and induced mutation, these are artificially caused by mutagens. So these agents are certain agents are there which has ability to induce mutation that are known as mutagen, and this mutagen causes a mutation, then it is known as induced mutation. So it is known that mutagen induce mutation at a at relatively very high rate, very high rate, and has made mutations, and it made mutation usable in plant breeding. Plant breeding. Now here the mutagen which cause the induced mutation, that mutagen are of two type. This mutagen are of two type, that is chemical mutagen and physical mutagen. Okay. Now if we talk about a chemical mutagen, then here chemicals like EMS, that is ethyl methane sulfonate, that is EMS, okay, and sodium azide are used to induce a chemical mutation. Induced mutation, there are chemical mutation. Now here in physical mutagen are radiations like gamma rays, UV rays, X-rays, etc., and that is going to cause a physical mutation. So mutation produced in response to the treatment with mutagen, it is known as induced mutation, and mutation breeding is the use of induced mutation in plant breeding to develop the improved variety. So by causing this mutation, we can develop a certain, we can say, the improved varieties. Okay, so here if we talk about the improved variety by the mutation, then we can say the Mexican wheat. We know that this Mexican wheat are Sonora 64 and Lerma Rojo 64. Now their improved variety by inducing mutation are Sarbati, Sonara, and the improved variety of Pusa Lerma Rosso 64 is Pusa Lerma. Such a way the varieties can be improved and which is made suitable for the what their growth or make them adaptable in our Indian environment. Now, apart from this, if we take some more examples of mutation breeding, then these examples are as following. And if we talk about the examples, the first is Raime Atomita 2 and Jagannath, varieties of rice. Second is Erectiferum and Erectoids variety of barley. The third is Aruna variety of castor. Fourth is NP 836 variety of wheat. And fifth is Indore 2 variety of cotton. These all varieties are developed by the mutational breeding, or that is we can say the induced mutation. So first is the conventional breeding, second is the we can say the mutational breeding. So these are the two things, that is conventional breeding and mutational breeding. Through this which we can get a disease resistance. But apart from this, other two ways are there through which we can gain a disease resistance. The third way is selection amongst somaclonal variants. And this selection amongst somaclonal variants, if we talk, then genetic variation present among plant cells during tissue culture is called somaclonal variation. And some of this may be useful and stable. So another disease resistance can be gained during a tissue culture through the somaclonal variation. And the fourth way of inducing the disease resistant in plant is the genetic engineering. So genetic engineering, if we talk, this is a process. This is a process in which one or more genes for disease resistance are taken out from other plant and incorporated in
cultivar.
Okay, so this is the process through one or more genes of a disease that are taken from the other plant and incorporated in the new plant or new cultivar. Then such a way the disease resistant can be given to the new plant. Okay, so these are the all ways how the disease resistance can be gained in the plant from the bacterial, fungal, and viral disorders. Okay, I hope you understood well, and that is all about the what a disease-resistant plant breeding for the disease-resistant. Now, in next lecture, we will be going to talk about the plant breeding for developing resistance to the insect and pest. Thank you.